My name is Melissa Mavers, and I am with Washington University in St. Louis. Research in my lab is focused on invariant natural killer T cells, which are innate lymphocytes that are called iNKT cells because they have variable surface expression of some of the same proteins as natural killer cells, but they also have a T cell receptor, and it's an invariant T cell receptor, meaning a defined alpha chain and beta chain. And because of the unique mechanism by which these cells are activated through that invariant T cell receptor, they have a lot of potential as a universal donor for cellular therapies. And so my lab is very interested in understanding the basic biology of these cells because much of the work has been done in mirroring iNKT cells. So we have several projects focused on studying the biology of human iNKT cells, trying to understand different functionally distinct subsets in the human cells. Also, we're focused on a project where we're trying to engineer iNKT cells to see if we can make them more immunosuppressive for the disease we're interested in studying, which is graft versus host disease. And so we have a third project in my lab with the goal of trying to understand the plasticity of iNKT subsets. And that project is using murine models and murine cells. iNKT cells are really important for potential clinical use because they cannot cause alloreactivity because of the mechanism by which they're activated. They could be a universal donor for cellular therapies. So they have a lot of potential for clinical translation into cellular therapies for cancer or even immunosuppressive cellular therapies. For example, as we're interested in graft versus host disease, which is a complication of hematopoietic stem cell transplantation. These cells have a lot of potential to be a cellular therapy for GVHD prevention or treatment. And so a lot of the work in our lab is very basic science, but also with a translational goal in mind. And so we are working towards better understanding how we can optimize use of these cells for a cellular therapy to help patients. I think the most interesting thing we've learned recently is that when we were studying the transcriptome of human iNKT cells, we found that there was a combined Th1, Th17 signature in some of the cells, which is different than what has been shown in murine iNKT cells, where these are very distinct populations. And so I think it'll be very important as we move forward in the work in my lab to investigate that and really understand what that means as we're working towards developing these cells as a translational goal for clinical use. I'm having a great time so far at AAI, and I think what I'm most excited about for the conference and most looking forward to as the conference progresses is a couple of things. First of all, meeting other people working on iNKT cells because they are a somewhat rare cell type, and so it's a small community, and I'm very interested in just talking about iNKT cells with people because I find them fascinating, and there are some other people who do as well, so I think that's going to be very fun. And then also I'm really looking forward to talking to trainees, just reaching out to the amazing junior scientists who are here and helping learn about what they're interested in, guiding them on their path, whether it ends up being with my lab or somewhere else. I would say you have made an amazing choice. Immunology is fascinating. No matter what aspect of it you pursue, I think all of it is very fascinating. And so I would encourage trainees to just continue pursuing it. Don't let anyone deter you from your goals, and find mentors, find good mentors and who will support you and who will sponsor you.